In the last videos, I talked about in vitro slice physiology, why it is an important and powerful technique to study the brain in action. And one problem is that there is a small slice of live brain tissue, and the challenge is to keep this piece of tissue alive for several hours after it has been extracted from the brain. And that is done by perfusing the tissue inside its holding chamber with a continuous stream of artificial cerebro-spinal fluid or brain juice. In some experiments, it is required to add very, very expensive pharmacological agents to manipulate the physiology of the slice in a particular way. And because those agents, pharmacological agents, can be so incredibly expensive, it is advisable to reduce the volume that is used in the experiment as much as the investigator possibly can in order to keep the costs reasonable. So scientists have come up with multiple ways to reduce the liquid amount required for a particular experiment, and typically can get the volume down to about 7 to 10 milliliters per experiment and per day. PopNeuron's in vitro slice chamber has reduced that required volume amount by a lot. Our chamber requires about 1.5 to 2 milliliters of solution, thereby again significantly reducing the cost of an experiment. And the way we were able to reduce the volume required for an experiment is by getting rid of any external holding containers and pipes and anything, because these external devices require to be filled with fluid as well in order to get a circulation going, and that is fluid which needs to be produced and needs to be paid for. So our chamber only has about 1.5 to 2 milliliters of ACSF directly in the recording chamber where the brain slice is located. And so how do we make sure that the solution in our chamber has enough oxygen and enough nutrients and does not accumulate toxins? So the chamber has an input for oxygen. So there is a little tube connected to the slice chamber that brings oxygen from an oxygen tank into that slice chamber, and the air pressure of that oxygen coming into the chamber creates a water pump action that continuously moves the brain juice in that recording chamber around and makes sure there are no pockets of stale solution created anywhere. In fact, the movement of solution in our chamber is a figure 8, and we tested this at first in modeling. So the chamber was designed in an iterative process where we put something together, tested it in a fluid model, and tweaked it until there was a solid continuous flow in a figure 8 pattern established without creating any pockets. Then we prototyped the chamber and we tested it with actual solution and actual gas, and then we put little red dots and dye into the chamber and took videos to make sure the flow of the solution in the real chamber is the same as it was modeled in the model before. And so we're very confident that the solution in our slice chamber is continuously oxygenated, is continuously moved around, and therefore can always provide the tissue with enough oxygen. How do we know the nutrients are enough, and how do we know there are no toxins being accumulated? Well, that was tested with liquid chromatography and mass spec. So we put a section of brain tissue in this chamber and left it in there for several hours, and we continuously took samples of the solution and we mass spec'd and chromatographied them to look for the accumulation of any waste products that could be dangerous, and we were also looking for a reduction in things like glucose. And the short answer to all this is there is not a significant accumulation of waste products. There is not a significant reduction of nutrients to be of any concern. The results are published in a paper which is linked on our website. So finally, as a last test, we used the slice chamber to do actual patch clamp recordings. So we put piece of brain tissue in the chamber, we patch clamped a cell, and we held it for as long as we could, which was an hour, 2 hours. Then we patch clamped the next cell and held it for as long as we could. And we looked for rundown in the physiological properties. And the short answer here is, again, that the rundown of physiological properties was not significantly higher than it would be under any other brain slice techniques, which use a lot more liquid fluid. And again, the results are published in our paper that are linked on the website if you want to go and dig into the details. Thank you.